Standard CRISPR-Cas9, while revolutionary, fundamentally relies on creating a double-strand break at the target locus. This break is the core limitation for precise sequence correction. The cell primarily repairs this double-strand break using non-homologous end joining, an error-prone pathway. Non-homologous end joining frequently results in random insertions or deletions, known as indels, which typically cause frame shifts and gene knockout. While homology-directed repair allows for precise edits using a donor template, this pathway is highly inefficient, particularly in post-mitotic or non-dividing cells like neurons. The inherent risk of large deletions or chromosomal rearrangements associated with double-strand breaks necessitates a safer, more precise editing tool for clinical applications. This challenge is precisely what prime editing was engineered to overcome. Prime editing is a gene editing technology specifically designed to avoid the formation of double-strand breaks by utilizing a fusion protein complex. This complex consists of a Cas9H840A Nickase enzyme fused directly to a reverse transcriptase enzyme. The Cas9 Nickase is engineered to cut only one strand of the DNA helix, preventing the catastrophic double-strand break. The key component guiding the precise edit is the prime editing guide RNA, or PEG RNA. The PEG RNA is significantly longer than standard single guide RNA because it contains 3 distinct functional domains. These domains include the spacer sequence, which is responsible for target recognition at the genomic locus. It also contains a primer binding site, and crucially, the desired edit sequence template itself. This template dictates the exact change to be incorporated into the genome. The prime editing complex first binds to the target DNA locus specified by the prime editing guide RNA spacer sequence. This binding occurs adjacent to the protospacer adjacent motif, or PAM sequence. The Cas9 Nickase component then creates a single-strand break, or nick, exclusively on the non-target strand of the DNA helix. This nick exposes a free 3' hydroxyland on the DNA strand. Following the nick, this exposed 3' end detaches from its complementary strand and subsequently hybridizes to the primer binding site region located on the prime editing guide RNA. This critical hybridization event is known as strand invasion or priming. Priming correctly positions the exposed DNA end, making it available for the subsequent synthesis step catalyzed by the reverse transcriptase. Following the priming step, the fused reverse transcriptase enzyme uses the prime editing guide RNA's edit template sequence to synthesize a new DNA segment. This synthesis extends the nicked DNA strand, incorporating the desired edit, which can be a substitution, a small insertion, or a deletion, directly into the target DNA. This process transiently creates a structure where the newly synthesized DNA flap is hybridized to the original complementary strand. Cellular repair mechanisms must resolve this intermediate structure. Enzymes like flap endonuclease 1, or FEN1, excise the original unedited DNA segment flap. DNA ligase then seals the remaining single-strand break, permanently incorporating the edit into one strand of the DNA helix. This mechanism ensures the precise rewrite of the genetic information without relying on error-prone double-strand break repair. The initial prime editing system, known as PE2, often suffers from low efficiency because the cell can use the unedited complementary strand as a template to repair the nick, reverting the intended edit. The PE3 system was developed to address this limitation by introducing a second guide RNA and a second nick. This second single guide RNA targets the unedited DNA strand downstream of the initial edit site. The resulting second nick biases the cellular repair machinery. By creating a second break, the cell is forced toward using the newly synthesized edited strand as the template for repair. This strategic introduction of a second nick significantly increases the overall editing efficiency and ensures a higher purity of the desired genetic outcome. Prime editing offers superior versatility compared to both base editors and standard CRISPR-Cas9. Base editors are restricted to only 4 transition mutations: cytosine to thymine, guanine to adenine, adenine to guanine, and thymine to cytosine. In contrast, prime editing can achieve all 12 possible base-to-base conversions. Furthermore, prime editing facilitates precise small insertions, up to 44 base pairs, and deletions, up to 80 base pairs, without relying on the error-prone homology-directed repair pathway or requiring a double-strand break. This broad scope allows for the correction of nearly 90% of known pathogenic point mutations. This includes direct rewriting of sequences responsible for diseases such as sickle cell anemia and cystic fibrosis, demonstrating unprecedented precision in genetic modification. In therapeutic development, prime editing is crucial for in vivo correction of genetic disorders, particularly in tissues composed of non-dividing cells, where homology-directed repair is ineffective. Delivery systems such as adeno-associated virus vectors are currently being optimized to package and deliver the relatively large prime editing machinery components to target organs like the liver or the brain.
In agriculture, prime editing enables precise modification of crop genomes to introduce beneficial traits, such as enhanced disease resistance or improved nutritional profiles. This level of precision allows researchers to optimize plant characteristics without introducing foreign DNA. This capability accelerates the development of next-generation microbial strains and resilient crops necessary for global food security.